We're gonna do a few quick Canvas updates and then we'll go into the PowerPoint. As a reminder, we don't have class Friday, it's Good Friday, so there won't be a WebEx, there won't be a video for you to watch. Enjoy your holiday if you celebrate. If you don't, do something fun. There we will pick up with chapter 7 next week. So let me show you what we have going on for Canvas. Okay, so we have, oh, very good. Okay, so this is your Canvas view. A few assignments I want to draw your attention to coming up. Click on the assignment tab. We've got your discussion questions for this week due on Friday at midnight. So that is nervous system questions part 2 that you need to answer, as well as participating in the things that I find interesting or confusing discussion board. So you need to do that for this week's credit for the discussion board. We also have the GLIA worksheet, which is basically just tell me about the 6 types of GLIA that we talked about. And that's due Friday at midnight. And then next week we have homework 5 that's due at the class time, so pay attention to that one. It's April 15th, due at normal class time, 10:10 a.m. You're gonna have another batch of questions for discussion and discussion boards for next week. And then you'll have at the end of next week a worksheet on the cranial nerves due, as well as a case study about monkey brains, which I thought was cool. So that's going to be finishing up the nervous system aspect, and then we'll be switching gears and we'll be looking at our senses, chapter 8. And as a reminder, we're not going to cover chapter 9 this semester. With losing a week and all the chaos with that, we just cut the endocrine system out. So we are about to approach our last chapter once we finish chapter 7. So with that being said, do people have any questions about what is due? Okay, so I'm going to switch gears. If you do, again, put them in the chat feature. I'm going to switch gears and we're going to jump to the PowerPoint. Okay. Switch to that. Okay, so we're going to pick up with the nervous system. We had left off, we talked about the parts of the brain last time. As a reminder, for parts of the brain, you need to know, for the 4 lobes, you need to know where those 4 lobes are. And that's simple because they're named for the bone that they're under. So your 4 lobes are your frontal, parietal, occipital, and temporal. FPOT, if you're looking for an acronym here, and they go with the cranial bone. So the frontal bone is with the frontal lobe, the parietal lobe on top is your parietal lobe, occipital lobe in the back is under the occipital bone, and temporal lobe on the side under your temporal bone. So you need to know where those are. You also need to know the functions associated with those lobes, which we went through. So if you see a list of functions, you have to be able to tell me where they go, or vice versa. If you see the lobes, you have to be able to tell me the functions associated with those lobes, okay? We then dove into some deeper brain structures, and for those, you don't need to be able to label them on any sort of brain diagram, but you do need to know what they do. So for the other structures, I care more about function than I do about location, okay? You got to be able to tell me what those brain regions that we talked about, what their job is, what their role is, okay? So we're going to switch gears and we're going to talk about this question that's kind of been an overarching question about neurons and why we protect neurons. We talked about it a little bit when we covered glia, especially when we're talking about the blood-brain barrier. But the main reason we need to protect neurons is they are post-mitotic cells, so they do not divide. So these cells, if they are damaged, they cannot replicate and replace the area. So when you cut your skin, for example, it's very easy for your skin to undergo mitosis and fill in the gap with new skin. Your neurons can't do that. If they are damaged and you lose neurons in an area like a brain damage or a spinal cord injury, that is it. You are done for that region, okay? The other reason that neurons are important, so not only are they post-mitotic and delicate in that regard, they also are really important. So the nervous system has this major integration role where it is responsible for taking in all the information around us and on our whole world and deciding what to do at both a conscious level and a subconscious level, determining all of our actions. So the nervous system is important, it's delicate, it uses a lot of energy, is the final thing to keep in mind with neurons. When we looked at the action potential, we talked about how ions, sodium and potassium and calcium at different parts, are moving in and out of the neuron. And all of that is based on their gradient. So again, ions are going to want to flow from an area of high concentration to an area of low concentration. And at rest, they can't get through the membrane. They can't do that. They need channels to be opened. And so in the neuron, we talked about these voltage-gated channels that are going to open up for sodium or potassium. And at the axon terminal, we talked about calcium. And the ions are going to flow from a high concentration gradient to a low concentration. And to put those ions into the right spot, we have to use pumps. So to make concentration gradients in our neurons that are required for action potentials, that are required for signaling, it takes a lot of energy. Sodium and potassium in particular, we can move those ions to their respective locations using the sodium potassium ATPase, so the sodium potassium pump. And each time you do that, you're going to use ATP. So your brain is going to need a lot of glucose to make a lot of ATP. So it has a high energy demand. So the things to keep in mind with neurons, they have this high energy demand, post-mitotic, so they can't repair themselves, and they have a super, super crucial, important function. And so because of that, we use 4 different systems to protect our central nervous system. We use 4 different systems to protect our central nervous system. One of them we already talked about, and we'll talk about it again in a little bit, is that blood-brain barrier. The other ones are bones. So we have around our brain, we have the skull, and around our spinal cord, we have our vertebrae. And we're not going to go into too much more detail in that because we covered it in our bone chapter. But keep in mind, we talked about all those different bones in our cranial and facial bones in particular that make up the skull. So all of these bony structures are protecting our central nervous system. The central nervous system also has layers of meninges that are going to be between the bone and the brain. And it's bathed in fluid. It's bathed in the cerebral spinal fluid that we abbreviate CSF. So the meninges help cover our central nervous system. If we start at the very top, we've got the skin of our scalp. That is going to be the very top layer. Under that is the periosteum, which is the layer that goes between the scalp and the bone. Then you're going to have that thick protective bone structure, right? We talked about functions of bones back in our bone chapter. Protection, the skull is a great example of that. Under the skull, you're going to see 3 layers shown here in blue of our meninges, the dura mater, the arachnoid mater, and the pia mater. You can use the acronym DAP or PAD if you go the other way. So the dura mater, the arachnoid matter, and the pia mater. And these are going to form that protective covering. And under each layer, you have sub-layers. So we talk about the area under the dura mater as our subdural space, the area under our arachnoid matter, our subarachnoid space. And then under that pia mater, that's where we're going to reach our CSF, okay? We'll have these little protrusions that come out in our subarachnoid space in particular. It's going to be where the CSF flows through. And there's going to be an exchange that occurs through the arachnoid layer of our CSF. Okay. So the CSF, or the cerebral spinal fluid, acts as a cushion. It's made from blood. It takes material from blood at our choroid plexus, and it's going to be located at the roof of your ventricles. So this is an anterior view of the brain. You can see that there are these holes in your brain, these ventricles. We have a lateral ventricle. We've got a 3rd ventricle you can see here, and the 4th ventricle going down. And these are going to be where the CSF is just going to move. So remember, we talked about glia. We have those epidermal cells that have the cilia on them. The cilia are just going to move the CSF through the ventricles and then down the spinal cord. And what this does is it acts as a cushion to kind of hold the brain in place. And so if you were to look at your brain, it would be like it was floating in a dish of fluid. That way, when your head moves around, the fluid helps it so that the brain doesn't move quite as much. It doesn't jostle as much because it's insulated with all this liquid, okay? So that CSF, if we look from the lateral view again, is going to move through the ventricles, like our lateral ventricles, into our 3rd and 4th ventricle, and then down into our spinal cord. And so this one is showing you the flow of CSF. So it's made at the choroid plexus at 1 here. It's going to flow through our ventricles. Some of it's going to go into that subarachnoid space, again, up near the skull. Some of it will enter the central canal of your spinal cord and will flow down. And then it will continue, the rest of it will continue through that subarachnoid space, where some of it will be absorbed back into the dural sinuses via those little arachnoid villi, so those little protrusions. And CSF is something that, for medical professionals, it's hard to get. So it's very easy to get blood, very easy to get blood from a few different places in your body to get a blood draw. CSF, you have to get through a lumbar puncture, or otherwise known as a spinal tap. So they have to stick a needle in between the vertebrae in your lumbar region and pull out some of that CSF. And the major reasons why they would do that was, one, to see what's going on. So if you analyze the contents of your CSF, it can tell you a little bit about what's happening in the brain. For example, it can tell you if you have a bacterial infection, if there's a high level of bacteria in that CSF. The other reason why you would get a lumbar puncture is to help relieve some of the pressure. So the brain is in this enclosed space with fluid, and if the brain starts to swell, it's going to push the fluid and increase the pressure because there's nowhere for that fluid to go. It's not like there's a leaky spot where it can just shoot up through your eyes or something. So instead, it will cause headaches and all sorts of other side effects as that intracranial, that brain pressure builds. And so one way to relieve that is to pull out some of that fluid through a lumbar puncture or through a spinal tap. Okay, so this brings us back to our fourth way we protect our central nervous system, which is through our blood-brain barrier, our BBB. Remember that the blood-brain barrier is made of astrocytes that kind of go around the capillaries with tight junctions in your brain. So tight junctions, not going to allow things to move between the cells. And this is important because then anything that wants to go through from the blood into the brain through the astrocyte has to use transport molecules. Keep in mind those very early contents that we talked about what is in your plasma membrane and what can move through the plasma membrane and what can't. So things that can move through your plasma membrane very easily are things like gases, so oxygen gas, carbon dioxide, very easy for it to move from the brain into the bloodstream. Things that are fat soluble, so small steroids, cholesterol, very easy to pass through, to sneak on through that plasma membrane. Everything else has to use a transporter. And so your brain has transporters for water. It has those aquaporins. It's got glucose transporters, and it's got amino acid transporters. But that's about it. So it doesn't allow large molecules, toxins, all sorts of things from your body to go from the bloodstream into the brain. Importantly, the only sugar it really takes up is glucose. So your brain needs straight glucose, which is why if you're hypoglycemic, if you've got low blood sugar, one of the first side effects is going to be like a lightheaded feeling and a dizziness and kind of a fogginess in your brain. And that's because your brain needs sugar in order to function. And it's got those transporters specifically designed to bring in and take in that glucose. Okay. This is why, as just a final note, as someone who worked on designing pharmaceuticals for the brain, it's a real challenge. So if you're going to take a drug and you want it to act in the brain for something like Alzheimer's or Parkinson's or all of these other brain disorders, mood disorders, it has to either be fat soluble or it has to be similar to an amino acid or to glucose in order for it to pass into the brain, because everything else is going to be stopped. And so this is a real challenge for people who are trying to design drugs that will affect the brain, is getting it through the blood-brain barrier, because it is that ultimate gatekeeper that separates the brain from the rest of the bloodstream in your body. Okay. So again, you need to think about those 4 ways we talked about that we protect our nervous system. We have bones, we have meninges, we've got cerebral spinal fluid, we've got the blood-brain barrier. And then you need to think about why it's so important that we protect our central nervous system. It's got a crucial role as that integration center, it's got a high energy demand, and they're post-mitotic. So they're not going to divide, they're very delicate. They need all of these layers of protection. Okay. In the chat feature, if you wanted to write down, if you have any questions so far, let me know. Otherwise, we're going to keep moving. for that. Okay. I'm going to keep going here. Let's talk a little bit about some brain dysfunctions. Again, things go wrong when we're talking about the brain, in part because they're post-mitotic. So neurons can't divide. If you damage them, it's a problem. One of the classic examples is a traumatic brain injury, or a TBI. What I'm showing here, great football team, football players and other people who get these repeated brain injuries, they can have long-term and high risk for developing a lot of disorders down the line because they're damaging their neurons, and the neurons can't replace themselves and they can't repair themselves very well. And so when we talk about symptoms of a TBI or symptoms of a traumatic brain injury, we have things like concussion versus a contusion. So a concussion is generally associated with a brief loss of consciousness, a brain injury associated with a brief loss of consciousness. And there's a whole guideline and checklist that people go through, especially related to athletes, to determine the severity of their concussion. Very severe concussions, you can be feeling side effects from that for months down the line, and they can damage or affect your learning, your memory, your cognitive abilities, whereas less severe concussions, you might be feeling okay within a few minutes. So concussions, again, associated with that brief loss of consciousness. Traumatic brain injuries also have contusions. This is a brain bruise, usually caused by an acceleration-deceleration event. So if your head is moving in one direction and then it stops and your skull stops, your brain is going to keep moving until it hits that skull. And that force of the brain, even with that protective fluid and all these like thin meningeal layers, your brain is still going to hit the skull with a certain amount of force. And if that is a high level of force, that's where you could get a contusion or a brain bruise. If it's a bad injury, you could also be looking at an intracranial hemorrhage, which is a brain bleed. Again, your brain is bathed in cerebral spinal fluid, and the blood is supposed to be kept in those tight capillaries surrounded by those astrocytes that form that barrier. And so the blood is not supposed to be moving around in the brain space at all. And when it does, it can displace a lot of the spinal fluid and lead to some other downstream bad side effects. And finally, we have a cerebral edema, which is a brain swelling. Again, if the brain starts to swell, it's a closed space, and so the fluid really has nowhere to go, and you could start to have issues of headache, and if it pushes on certain regions, you can get blindness, you can get other side effects there. And the way that they relieve that is they're going to take a needle and they're going to pull out some of that cerebral spinal fluid from your spinal cord and relieve some of that pressure. Okay. Traumatic brain injuries. Another brain dysfunction that I want to talk about are strokes. They're currently the third leading cause of death in the United States. And strokes, the medical term for it is going to be a cerebrovascular accident, or a CVA. And symptoms of a stroke depend on where the stroke occurred. So the stroke is caused by a blood clot in one of the capillaries in your brain. And it will cause dysfunction based on what that area of the brain does. So if you have a blood clot in your motor areas, you're going to lead to paralysis. This can be usually a hemiplegia, or one half of the body will be affected. If you have a blood clot on the left side of your brain in a motor area, the right side of your body will go numb, you'll be unable to move the right side of your body specifically. And if you have it on the right side of your brain, the left side of your body, you'll be unable to move that side of your body. As we talked about with Broca and Wernicke's area, you can have aphasias if the blood clot is in a language area, again, a language area on your dominant side. And then all of this is a less severe stroke we call just a transient ischemic attack. So again, it's caused by that blood clot in the brain. And if the blood clot is small, your body will be able to push it out and you won't have very severe side effects. If it's not, if it stays there, it can start to kill the tissue right around that blood clot because it's not getting oxygen specifically. It's not getting that really crucial gas. Also, it can burst, and that's what people really worry about, is when the transient ischemic attack causes an intracranial hemorrhage or a hemorrhagic stroke. Those are when you're talking about some really bad outcomes. And so if you see someone and you suspect that they're having a stroke, you need to get them to medical attention really, really quickly. Time is of the essence, because drugs that doctors can give that will help dislodge that blood clot and help bring oxygen back to the area. But the faster it happens, the better the outcome will be. So if you notice someone who has drooping specifically of their face on one half, or one part of their arm is going numb or their leg is going numb, they start to have issues with speech, whether that is disordered speech or speech slurring. They start to feel confusion and a general fog. You want to get them to the emergency room really as fast as possible. Okay. We're going to switch gears and move from the brain to the other part of our central nervous system, which is our spinal cord. So the spinal cord is our information highway. It is about 17 inches long. It goes from your skull to the lumbar region, L1, L2 approximately. And like your brain, it's covered in meninges and it's covered in bone. Here, the bone is the vertebrae. And shooting out from your spine, you have 31 pairs of spinal nerves, which have sensory and motor nerves from it that are going to innervate or give feeling and motor movement to your body. So your 31 spinal nerves come out from your spine and turn on all of the muscles and give all the sensory input to your body. At the very end of the spinal cord, the nerves that come out the bottom end, the inferior end, we call the caudae equina nerves. And this comes from equina meaning horse. If you've ever been horseback riding, you'll notice that, and this is all secondhand, I've never been on a horse, but I've been told if you go horseback riding, the inner part of your legs is the area that's going to get sore first just from being on the horse. Well, the nerves at the inferior part of your spinal cord, they innervate or they turn on the inner part of your leg. And so if you get damage to the caudae equina region, then you're going to be numb and unable to move the inner part of your leg. And so that's where the name comes from. So caudae equina region, inner part of your legs, horseback riding muscles. Okay, so this is what the spinal cord looks like. Again, the nerves are named for the region that they exit the spinal cord. So our cervical spinal nerves exit via the cervical vertebrae, and so on with the thoracic, the lumbar, and again, it ends at the lumbar region. The end of the spinal cord is in that lumbar region, and we will have the caudae equina nerves coming out, shooting down from there. And the spinal cord is enlarged, it's thicker in two key regions. You have a cervical enlargement and a lumbar enlargement. And these enlargements are because you're going to be sending out a lot of nerves to innervate your arms at the cervical enlargement and your legs at the lumbar enlargement. So they're bigger because they need to accommodate all of the nerves that need to go to these large body regions. Okay. Your spinal cord has both motor and sensory information in the gray matter. So if you look down here on the bottom, it's showing you a slice of your spinal cord. We've got the white matter. Again, those are the myelinated bits, and you have the gray matter here. And the gray matter we break into a dorsal horn, which is the back region, and a ventral horn, which is that front region. In the dorsal horn, we're going to have our sensory neurons. So the dorsal horn is going to have the neurons that come from the body on that afferent path and are going to go up the spinal cord to the brain, giving information about what we're feeling, touching, all of the temperature, the sensory information. The ventral horn, the part in the front, is going to be the motor nerves. So this is going to be coming back down from the brain to our muscle saying, contract this muscle, contract that muscle, coming from the ventral horn. So in the spinal cord themselves, this information is separated. We have motor on one side, sensory on the back, on the other side of our spinal cord. They will shoot out processes from those horns, and those nerves will then fuse into our spinal nerves. So your spinal nerve has both the sensory nerve and the motor nerve wrapped together. But when they come to the spinal cord, we will separate that information. We'll put sensory in the front and motor in the back. What this means too is if you get a spinal cord injury that is not fully bisecting. So if your spinal cord injury just damages a part of your spinal cord and not the whole thing, then you might have some interesting or different side effects depending on what side of the spinal cord is injured. So if a person injures the dorsal part of their spinal cord, the back part, they might lose sensation, but still be able to move those muscles. And the damage is going to be at the area, everything below loses input because they're not able to send the signal to the brain. So for example, if you get damage at the lumbar region, for just the dorsal horn, you would be unable to feel your legs. You would have no sensory input at your legs, but you would be able to move them. The motor part would be intact, and vice versa. If you get damage at the ventral side, you might be unable to move your legs, but you could still feel them. And again, for the spinal cord, it's damage at the level, and then everything below is unable to send that information up. So if you're damaged all the way up at the cervical level, that's where patients are unable to move their whole body, so arms and legs. Where if, again, if it's just the lumbar region, the arms are still going to be functional. It would be the legs that are affected. And this is what that looks like. Again, in the spinal cord, we separate out our sensory and our motor information, and they come together to form our 31 spinal nerves. Again, sensory is that afferent tract, motor is an efferent tract. Make sure you keep those vocab words straight. Sensory, afferent, which approaches the brain. Motor, efferent, exits the brain. Okay. This is what it all looks like together. So we start at our sensory receptor that's going to send information to the spinal cord on that afferent pathway. The cell body here is located outside the spinal cord. That information is then going to go up through the spinal cord, pass through the brainstem, and go up to give a response. The response will come from our sensory region in our brain, and then it'll pass that information to our motor region, right? Keep in mind where those are. You need to know different lobes that those occur in, which will then send the information down. The thalamus, again, is that relay station. So the information will go down and then become a motor or efferent neuron that will have a response. The other thing I want you to see here is that the information will cross. So information about the left side of your body is sent to the right side of your brain, and information about the right side of your body is sent to the left side of your brain. So there is this crossing that occurs right before it reaches the cerebral regions of your brain. Okay, questions you need to consider. So we're going to briefly talk about the peripheral nervous system. For this section, we'll talk about the structure of a nerve, and then we'll focus on our cranial nerves and talk about the sympathetic and parasympathetic system. So again, keep in mind, a nerve is a bundle of fibers when we're talking about the peripheral nervous system. Our spinal nerves are called mixed nerves because they have both sensory and motor information. Once they leave the spinal cord, remember our spinal nerves fuse, they have sensory and motor information. There are other nerves in your body that we'll talk about that are sensory and motor only. Okay, so today we talked a lot about brain dysfunction, we talked about spinal nerves, and we spent the first part talking about the ways that we protect our central nervous system. So those are the key things I want you to take away from this unit that we just discussed. We have those things that are going to be due on Canvas, and we won't have class Friday because it is the holiday. I'm going to stop the recording here.